microbes, and we exist only to the extent that we can hold those microbes at bay. Dr. Shirong Liu was the first to discover functional microRNAs in the mammalian gut lumen and feces. He's interested in host microbe interactions, particularly how the host evolves with the symbionts in and around it, how the symbionts were specifically selected for specific hosts, and what the role of that symbiosis is in autoimmune and neurodegenerative diseases. Following Dr. Liu today, we'll have Dr. Christine Faherty. She's enhancing our understanding of gastrointestinal bacterial pathogens, particularly Shigella, with the goal of identifying vaccine candidates that could improve the health of millions around the globe. And then because bacteria aren't the only things we have to deal with, Dr. Damien Kreisen is a pediatrician who's interested in understanding how pathogenic fungi cause disease, how the host defends itself against these fungal pathogens, and how to develop new therapies. His work currently focuses on some of the most common fungal causes of human infection, including Candida albicans, which arguably is a host commensal, a human commensal, and Cryptococcus neoformans and Aspergillus fumigatus that arguably are environmental fungi capable of causing severe human disease. Interspersed between those talks, again, we'll have a fantastic opportunity to enjoy the art and hear more about the scientists, artists, including Dr. David Goodsell and Dr. Anna Maria Porras. And then again, like the first session, we'll finish with a 45-minute question and answer period. So if you don't get your questions answered between talks, we'll have plenty of time to discuss the material then. So it's a pleasure today to have Dr. Shirong Liu. And so Shirong, let me turn it over to you then.
Thank you, Mike, for your introduction. And hello, everybody. Thank you for joining us to discuss about the host microbe and interaction. This is the, so just a second, let me share the screen. Hello, can everybody see my screen?
That looks good.
Okay. Hi, everybody. Hope you enjoy your lunch and help your bugs in your gut happy. So then I'm going to talk about the interaction between the host and the commensals in the gut, particularly how the microRNAs that we found in the gut involved in this interaction. So my talk will be separated to two parts. The first part, I'm going to, in general, talk about the interactions between the host and the microbe mediated by the microRNAs. And then the second part, I will talk about the specific microRNA. How does this microRNA involved in the specific disease, multiple sclerosis? So as you know, everybody know nowadays that the commensal play a really important role in the health maintenance and involved in disease pathogenesis. So I think it's very, very important to keep the commensals in shape. A very important question at least in my mind, is how these microbes, because the microbe and the host is really specifically really specific. The microbe is really specific for the host, and such relationship is so specific that I think that there should be a chosen selection mechanism that the host can select a specific microbe to the to the host. So my background is in immunology. So I think about this question, whether this the microbes is selected by the innate immunity, adaptive immunity, or something else. So when I look at the literature, I found that actually, if you look at the microbiome data in the mediated knockout mice, this mice actually is mediated is toll-like receptor signaling adapter. So in this mice, basically they knock out the innate signaling, and you can see. Let me just put here the laser point. So here's what the purple ones are white type and red ones are mediated knockout mice, and you still find that they're clustered to each other. So that means they have a really cool core microbiome. It's not something like the loose core microbiome. And if you look at the red one knockout mice, these mice are deficient of adaptive immunity. They don't have a mature T cell and B cells. In these mice, you can also see the core microbiome in the same group. That means adaptive immunity is not really dominate the factor to shape the gut microbiome. I show here just an additional evidence from the literature. Chief is another adaptive molecules in the toll-like receptor signaling pathway. It involved in signaling through downstream toll-like receptor 3 and toll-like receptor 4. And in this double knockout, mediated and achieved double knockout, you still find a core microbiome. And this is just to repeat another additional evidence saying that in the adaptive immunity knockout mice, the microbiome is shifted, but as you can see, it's still clustered to each other. They didn't lose the core microbiome. So we find something interesting in the gut when we look at the gut. When I joined the lab of Dr. Holdweiner's lab, his group were working on the circulating microRNA. So my colleague was looking at the microRNAs in the blood. So I joined the lab to work on the microbiome. So I asked the one question: so if the microRNA can exist in the serum and plasma, why can it exist in the stool? So we look at the stool samples and found a lot of microRNA there. You just show the top 10 and 15 microRNAs both in the mouse feces and the human feces. These microRNAs come from the gut epithelial cells, because if we generated the intestinal epithelial cell specific of dicer knockout mice. Dicer is an enzyme that is really important in generate the microRNA. If you knock out this dicer in the gut epithelial cells, you find that the microRNA in the feces are dramatically dropped. Those microRNAs also can come from the Hobbs positive cells. The Hobbs positive cells divided to the panel cells and the goblet cells in the gut. If you knock out the dicer in these cells, you can also find the geometric drop of the microRNA. But if you look at the microRNA in the red wine knockout mice, this means that the mice are deficient of mature T cell B cells, and you can find a few microRNA changes, but it's not universally dropped. That means most of these microRNAs come from the gut epithelial cells. So, what's the relationship with between the gut between this microRNA and the gut microbes? we look at the microbiota profile in this gut epithelial cell Dyson knockout mice, we found that the wild type, in the wild type mice, you can really find a really nice clustering of the of the microbiome profile. But in knockout mice, they're just randomly distributed. They're not clustered. That means the loose core microbiome. If you look at the similarity of the microbiome, you can see in the knockout mice that just don't each mouse is different from the other. So in the phenotype, we want to have a look. Okay, so if the microbiome is changed upon knockout the microRNA in the gut, so what's the consequence? We use DSS-induced colitis model. We induced colitis in this mice. It found that upon the gut epithelial cell microRNA deficient, and the disease is much more severe, as you can see in the blue one. Interestingly, if you isolate the microRNA from the feces and transfer to the knockout mice, then you can partially recover the microbiome to be more similar to a wild type mice. And when you induce colitis in this recipient mice, the recipient that received the fecal microRNA from the wild type mice, the disease is less severe. So this is kind of interesting. So that means the microRNA is involved in shaping the microbiome. Specifically, can we show that this microRNA can change specific microbe? We get some microbes as an example and some microRNA. We synthesize the microRNA and put in the culture of the bacteria. We can find that this microRNA can get into the bacteria. Here is we use a GFP labeled E. coli. We find that the localization of the microRNA with the bacteria. Here is just another tool to look at the microRNA in the bacteria using the flow cytometry. So how does this work? We compare the sequence of the microRNA versus the gene of the bacteria. We found some microRNA can really well pair the bacteria gene, and then we synthesize the microRNA and culture with the bacteria, and we find that this microRNA can change the growth of the bacteria. Here's an example of Fusobacteria, and then another example of the E. coli. And this is seen to be sequence specific because this one, so this mere 515 can only increase the Fusobacteria. It can not increase the abundance of E. coli, and vice versa too. And the reason, I think, is because the microRNA can regulate gene expression, as we show here. This particular microRNA can increase the gene expression of 16S RNA in the Fusobacteria, and this particular microRNA can increase the YGH in the E. coli. So come back to the question. So I just was curious of which system is more important, innate immunity, adaptive immunity, or here the microRNA in the gut. So we use the infectious bacterial infection model. We infect the mice with the Citrobacter. We compare the mice of the wild type B6 mice or the wild type litter mice of the gut epithelial cell and Dyson knockout mice. We also compare to the REG1 knockout mice and mediated knockout mice. We found the Dyson epithelial cell knockout mice is the first one, the most severe one, because it really died out before like 10 days after infection. And the other mice, the mediated knockout mice is secondary severe. The red one knockout mice is first severe, and the other mice can totally recover after a while. So this evidence suggests that we already identified the microRNA in the gut, and we show that these fecal microRNA are mainly produced by the gut epithelial cells, and this microRNA is able to get into the bacteria, specifically regulate the gene expression of the bacteria and affect the growth of the bacteria. And we show that by transfer the microRNA to the recipient, you can change the microbiome. So we conclude that this fecal microRNA can shape the gut microbiome. Okay, now we go to the second part. The second part, I'm going to talk about the specific microRNA that we found in the gut of the multiple sclerosis patient, and also in the mouse model of MS-EAE. When we look at the microRNA profile by sequencing, we found a specific microRNA miR30 that is increased in the EAE model as well as increased in the MS patient. And this is something interesting because when we synthesize this microRNA and give to the mice, give to the animal model of the MS, we found this miR30 can improve the disease. And when we treat the mice with antibiotics, while we treat the mice when we treat them with miR30, we found that this phenotype disappeared. That means the miR30 is working through regulate the microbiome. This actually is also evidenced by the fecal transplantation. When we take out the feces from the miR30 treated mice and give to the antibiotics pre-treated mice and then induce EAE, we found that those recipients received the fecal samples from the miR30 treated mice, treated donors. The disease is less severe. So then this suggests some microbe in the feces mediated the effect. So, and then which bacteria is mediated this effect? Then we look at the microbiome changes in the MS patient as well as in the EAE model. As we published previously that the specific bacteria named Akkermansia muciniphila is increased in the untreated MS patient. And this is just the qPCR data. Also, another cohort also showed that is increased in the MS patient. And when we look at the animal model, we also find Akkermansia was increased in the peak EAE. So we asked whether this particular microRNA miR30 can regulate Akkermansia. First, we look at whether this microRNA can get into the bacteria, which, as we show here with the electron microscopy result, this microRNA do get into the bacteria, and it can, by informative analysis, show that this potentially can regulate this particular gene in the Akkermansia. As we show here through the qPCR, it do upregulate the gene expression of this particular gene. And then when we culture this Akkermansia with method D, it can increase the abundance of Akkermansia. We give this particular microRNA to the mice and then profile the microbiome. We didn't see the shift of the general microbiome profile, but we do see it increased Akkermansia, particularly Akkermansia. And this is just qPCR data. So it seems method D increased Akkermansia, and through this change the phenotype of EAE. So the next question is, does Akkermansia really improve EAE? Then we did the experiment, and then we induce EAE and then treat these mice with Akkermansia. We found that Akkermansia do reduce the severe of the disease. How does this work? So we observed that in the Akkermansia treated mice, the regular T cells were upregulated. And then we do the in vitro culture. So how does this microbe Akkermansia upregulate Tregs? We culture the bacteria with naive T cells and trigger them to develop to Tregs, TF17, TF1. We didn't see it change TF1, TF17, but we see that upon co-culture with dendritic cells, Akkermansia can increase Tregs. We didn't see it by pure culture, the naive T cells, it cannot upregulate. Only when we co-culture with dendritic cells, it can upregulate. That means dendritic cells is involved in recognize or in response to Akkermansia stimulation. And then when we look at the dendritic cells, what changed upon Akkermansia stimulation, we found it can increase the secretion of TGF-beta and secretes much less IL-6 and less IL-1. These cytokines are important in the differentiation of T cells, and with less IL-6 and with high concentration of TGF-beta, you get Tregs. So this, I think, is a mechanism. So here I just want to summarize the second part. We found the microRNA method D is increased in the feces of peak EAE and untreated MS patient. And we found that the synthesized method D, by giving orally, it can improve EAE in the microbiome dependent way. We found method D can enter the Akkermansia, upregulate expression of the lactase in Akkermansia, and expanded the bacteria in the gut. And Akkermansia can expand the phosphate and positive Tregs, and then improve EAE. Okay, that's the story. I would like to acknowledge my colleagues, Dr. Weiner support the whole study, and a lot of colleagues in very helpful, help doing the experiment or analyze the data. Thank you for your attention.
Terrific, Dr. Liu. That was fascinating and really stimulating. Just to remind the audience, EAE stands for experimental autoimmune. And then what?
Encephalitis.
Myelitis, encephalomyelitis. And your readout for that is what?
We read out this clinical score. Sorry, it's a clinical score. Yeah. We also show those data in the histologic data, but when we present, we just put here and EAE scores, clinical score. The higher the score, that means the disease is more severe.
So really fascinating that these microRNAs somehow look to be involved in not just as a consequence of disease, but in the causation of disease somehow in an autoimmune disease. And they seem to do so by regulating the composition of members of the microbiota.
Yeah. What we found there actually very interesting is we found, so in the gut microbiome profiling study, including ours and a lot of others, cohorts show that commensal actually is more abundant. They found more abundant in the patient, in the disease patient. So people were thinking probably this is a pathogen induced MS or induced in the mice induced EAE, but actually we found it's not. It's just it's a protective mechanism so that a patient, when one get a disease, he or she is going to get like a response. And in this case, most likely it can generate like a miR3D, and this microRNA can activate these bacteria, and these bacteria play a protective role. These bacteria can just come to recover the disease.
And Amanda asked a question that was crossing my mind as well. So how do these microRNAs then get taken up by bacteria?
The mechanism how they take up the bacteria take up the microRNA, we still don't know. We are working on that. I think it's in general is something like so we know there are a lot of horizontal gene transfer, and probably it's a similar mechanism, but we don't know at the moment.
Fascinating. And do you have any sense for the relative stoichiometry of the amount of microRNAs versus the number of bacteria in proximity to the epithelial boundary to know how saturating is the availability of those microRNAs?
This, you mean the ratios or how about this? Actually, I don't know, but what I can say is the abundance of the microRNA in the feces or in the gut lumen is very, very high. It's much more higher than those you can find in the blood, in the serum and the plasma. But the question you're asking is what the ratio of whether the 1 to 10 or 100 ratio regarding the gut epithelial cell number, the gut bacteria cell number, or in the microRNA number, and we didn't quantify that. I think it's kind of hard because we can say, okay, we can say how many nanograms or picograms of microRNA we can find in a particular weight of feces or a weight of gut lumen content, but we cannot say the exact number of the microRNA.
Sure. And one other question, a couple of other questions, but one is we talked about how they that we don't know much about how they get into the microbial cell. How do they get out of the epithelial cells?
We do look at them. So actually, this is related to the protection mechanism, how this microRNA exists in the gut. We look at the microvesicles in the feces in the gut lumen. We found a lot of microvesicles of different sizes. And we found that this microRNA, particular microRNA, a lot of them actually can exist in the microvesicles. Most of them is in the microvesicles of size in around 100 nanometer. And we also find a lot of them actually they're not within the microvesicle, they just can exist in a vesicle-free state. So I think some of them is secreted from the gut epithelial cells in the form of the microvesicles. Others probably just release, I don't know in which form, or because when we profile the microvesicles, we use a size filtered approach. We just go down to 20 nanometer, the size of 20 nanometer, and then below 20 nanometer, we say this is vesicle free, but we are not sure whether it's really vesicle free. But yeah, that's what
Great.
I think. Yeah.
And then just in the 1 second or a few minutes, few seconds we have left, is there any known role for these microRNAs? This is a question from Hyman Hartman. Any known role for the microRNAs in the epithelial or other cells themselves, host cells?
Yes. Of course, microRNA endogenously exist in the host cells, yeah. But they are playing different role. Yes, and most of them. But
for example, the one that affects Akkermansia. Well, what's known about its role in the cell? Anything or?
Yes, in the circulation, actually, there are already paper talking about this microRNA and the target in the in the heart tissue, in the cardiovascular side. Sorry, but I cannot. It's like a muscle tissue in the heart.
Okay.
The cardiomyocytes, I think.
Yeah, that is yeah. Thank you. So they can regulate the proliferation of these cells. I didn't see it can directly change the behavior of the gut epithelial cells. I didn't see any literature talking about that. But in the case, well, this is if we come back to a general state, we found some of those microRNAs specifically secrete to the gut lumen. They are not they have really high concentration in the gut lumen, but much less concentration in the gut epithelial cells. That means they're by purpose secreted to the gut to do something.
Great. Well, thank you again. That was really quite stimulating and a very fresh and interesting perspective on the dynamic between microbes and the host. So thank you again.
Thank you.
With that, Hannah, let me turn it over to you to tell us